Hello, my name is Rachel Schafer. I'm a graduate student in Dr. Daniela Salvemini's lab, and today I'll be talking to you about CART peptide GPR160 induces ERK phosphorylation in astrocytes. So our lab studies neuropathic pain. This is a type of chronic pain that results from damage to the nervous system even after the injury is gone. And the goal of our lab is to identify novel therapeutic targets that can be used to treat chronic pain instead of opioids. One of these potential therapeutic targets we identified is GPR160, as we have found is a major determinant in neuropathic pain. We also identified the ligand for this receptor, which is CART peptide, and CART is involved in many physiological processes and pathological states, including pain. And we found that it evoked pain hypersensitivity in rodents through GPR160-dependent ERK phosphorylation. This leads to our hypothesis that CART peptide activates GPR160, leading to ERK phosphorylation, specifically in astrocytes. We chose astrocytes as they express high levels of GPR160 and they are major determinants in neuropathic pain. So first we want to see if CART activates the ERK pathway in our immortalized astrocyte cell line. So we treat the cells either with vehicle or CART at different time points, and we look at protein expression levels of phospho-ERK over total ERK. And at 15 minutes, we can see that CART induced ERK phosphorylation. However, by 30 minutes, it decreased. And next, we want to see if CART activates this pathway by either treating the cells with vehicle and CART or a MEK inhibitor and CART. MEK is a kinase that is upstream of ERK and it phosphorylates ERK. So if you block this kinase, you either decrease or block ERK phosphorylation. And when we co-treat the cells with vehicle and CART, we get an increase in ERK phosphorylation. However, co-treatment with CART and the MEK inhibitor, we get no response, indicating that CART does activate the ERK pathway in astrocytes. Finally, we want to see if GPR160 is required for a CART-induced phosphorylation of ERK. So we co-treat the cells either with CART and a IgG control antibody, which results in increase in ERK phosphorylation. However, when we co-treat the cells with CART and a GPR160 neutralizing antibody, we get a decrease, indicating that GPR160 is required for CART-induced phosphorylation in astrocytes. The significance is that understanding the signaling pathway can provide insight on CART peptide GPR160's functional role in astrocytes, especially since they are emerging as key players in neuropathic pain. And we also want to target GPR160 as a novel approach for treating chronic pain. So in order to develop a small molecule inhibitor, we have to understand the signaling pathway, and we could also use that signaling pathway to design a high throughput screening method. I want to thank the center, my mentor, the Salvemini lab, and the funding.